Welcome back to Acing AP Biology. In this lesson, we will continue our discussion of viruses by exploring animal and plant viruses. Animal viruses are coated with a membrane envelope. This membrane envelope contains glycoproteins that bind to receptors on the host cell. The viral membrane merges with the plasma membrane, allowing the capsid to enter the cell. Inside the cell, the virus removes the capsid coat and utilizes the cell machinery to reproduce its genome and proteins. As the virus leaves the cell, it takes on a membrane envelope stemming from the host plasma membrane. Some viruses are latent because they incorporate their genome into the host cell's DNA, becoming a provirus, similar to a prophage like with bacteriophages. These viruses remain latent until they respond to a certain stimuli. The symptoms resulting from viral infection can be due to the toxins secreted by infected cells, toxins from the virus itself, cells destroyed by the virus, and the body's immune response against the virus. Emerging virus is a term used to describe a new virus in the human population. It may be caused by a mutation in a current virus, a species jump of the virus from one species to another, or the spread of the virus to a larger population. Now let's go into the general animal virus life cycle. First, the glycoprotein on the virus binds to a specific receptor on the host cell. Then the virus enters and removes its capsid, exposing the viral genome. Some of the viral DNA is used for replication of this DNA, utilizing the host cell's enzymes, like DNA polymerase, for this process. Some of the viral DNA is also used for transcription and translation of viral proteins. The virus uses the host cell's enzymes and ribosomes for this process. Once the capsid proteins are produced and viral DNA is replicated, self-assembly of the new virus occurs. The new virus then merges with the host cell membrane to exit the cell, and takes on a membrane envelope derived from the host plasma membrane. It can now infect other cells. Now that we've discussed general animal viruses, let's look into retroviruses. Retroviruses are RNA viruses. The viral RNA is first transcribed into complementary DNA or cDNA by reverse transcriptase, which is an enzyme carried by the virus. The cDNA is incorporated into the host DNA, and then it is transcribed by the host cell, forming RNA. This RNA serves as the new viral genome, but also mRNA that can be translated into viral proteins. So RNA is converted to DNA, then to RNA, then to protein in the retrovirus life cycle. HIV is an example of the retrovirus. The membrane envelope for HIV carries glycoproteins that can bind to the receptors on T cells, which are essential for the immune system. It consists of 2 strands of RNA enclosed in a capsid, as well as reverse transcriptase. Let's now go over the retrovirus life cycle in more detail, focusing specifically on HIV. First, the HIV virus binds to the host cell. Specifically, the glycoprotein binds to the CD4 receptor on the T cell. It then enters the cell and removes its capsid. The viral RNA is then converted to complementary DNA using reverse transcriptase. The cDNA moves into the nucleus and incorporates itself into the host DNA, becoming a provirus. HIV can stay latent for a long time. Certain stimuli induce the transcription of cDNA into mRNA. This mRNA acts as both the viral genome and an mRNA transcript for translation of viral proteins. Then the virus self-assembles and leaves the host cell, taking on a new membrane envelope from the host cell. Now let's discuss plant viruses. The majority of plant viruses are RNA viruses. Plant viruses can dissipate through horizontal or vertical transmission. Damage to plants heighten their risk of viral infection. Insects may act as a vehicle for plant virus transmission. Viruses move from cell to cell via the plasmodesmata. Lastly, let's talk about some infectious particles that are even more rudimentary than viruses. Viroids are small, circular RNA molecules that infect plants and hinder metabolism, thereby weakening plant growth. Prions are small proteins that transform normal proteins to abnormally shaped proteins. Prions can cause mad cow disease. That's it for this lesson. If you have any questions, feel free to comment below. See you next time. For more bio content, hit subscribe.